Hello, 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 Mr. Sir. Hello, hello, hello.
Hello.
Hello, sir.
Hello, sir.
May I audible? Hello.
Hello, कहिए Mesh.
Sir. Please share your screen, sir.
Yeah, okay.
Okay, it's visible. Okay. Now we start. Hello.
Hello.
Good morning, everyone. Myself, Dr. Mahesh Kamkar, Program Coordinator of this online training program on emerging urban farming technology for vegetable production on the behalf of Center for Advanced Agriculture, Science and Technology for Climate Smart Agriculture and Water Management, Mahatma Phule Krishi Vidyapith, Rahuri. On this occasion, I would like to welcome today speaker Dr. Raghavendra K. Mehta sir. Welcome sir.
Thank you, sir.
Just I introduce this Mehta sir, a professor and head Department of Plant Pathology, University of Horticulture Science, Bagalkot. He is having educational qualification MSc Agriculture and PhD Plant Pathology with teaching and research experience last 23 years. He is specialized in plant pathology, research work in sunflower, safflower, chili, onion, tomato. He is awarded with Best Research Scientist from University of Horticulture Science, Bagalkot. Besides this, the job involves coordinating student academic activity, conducting as well as convening the research activity and addressing farmer problems mainly with related to horticulture crop. Thank you. Welcome, sir. And third session topic is integrated disease management of vegetable under protected cultivation. And also welcome, I would also welcome to Dr. J.S. Patpute, Madam, Organizing Secretary of this training program and co-principal investigator of CAST project, Department of Agriculture Engineering, Mahatma Phule Krishi Vidyapith, Rahuri, College of Agriculture, Pune. I would also welcome to Dr. Sunil Gorantiwar, Head Department of Agriculture Engineering, Mahatma Phule Krishi Vidyapith, Rahuri, and Principal Investigator and Convener of this training program. And I would also welcome to Dr. S.D. Masakar, Sir, Associate Dean, College of Agriculture, Pune, and welcome to Dr. Mukund Singh, Sir, Co-Principal Investigator, CAST Project, MTKV, Rahuri. Welcome to all participants, CAST team members, farmers, and all CAST research associates. Now I would like to request to today eminent speaker, Dr. Mista sir, to start the session, please. Please, sir, continue.
Thank you, Dr. Mahesh, for your nice introduction. Am I audible?
Yes, sir, audible. Audible,
Okay.
sir.
Okay.
Audible.
Fine. Thank you. So, respected dignitaries, all the organizers, I'm very happy to deliver my talk on integrated disease management of vegetable crops under protected cultivation. In fact, this very training program is with respect to urban cultivation, but some exotic crops, vegetables also I'm going to cover it, and mainly in the protected cultivation, the vegetable crops. My presentation runs the time allotted here is 1 hour, 1.5 hours, and my presentation runs almost 50 minutes to 60 minutes, and we can have discussion immediately after the presentation also. In between, if you have any problems, you can interrupt me as well. Let me start. So, as they have introduced, as Mahesh has introduced, I'm Dr. Raghavendra Mista, Professor and Head, Department of Plant Pathology at the College of Horticulture in the University of Horticultural Sciences, Bagalkot in Karnataka. Uh, so, let us begin with the concept of diseases. So maybe all of you know what is a disease. A very simple meaning disease is dis-ease. If you are not ease, if you are not well, it is disease, simply called. So disease means you are not well. This is a very simple English meaning of that. But normally in plant pathology we define disease in a scientific manner. As such, maybe you have, if you have studied during your undergraduate programs, if you are a BSc graduate or in some other books also, disease is normally deviation from normal functioning of the organism, normal functioning, deviation from the normal functioning, and it is mediated by a living organism. So that's most important here, living organism. See, what does it mean? Deviation from normal function. So you are, all the organisms in this universe, including plants, animals, everything, they are supposed to function normally, all physiological and biochemical activities. But if there is any deviation, and this deviation, if it is through the some living organism like bacteria, fungi, nematodes, viruses, so like this, many organisms. If there is any deviation, then we call it as a disease. But still, and this definition is still again modified, and many a times we use a more technical definition, or rather, we say that disease is actually what you can say interaction between three factors. Interaction between three factors, and it is well represented by a disease triangle. We normally tell that disease triangle. Disease is a triangular shape. That means to say it is having three sides. So three sides together, if they join, then only there will be disease. What are those three sides? One side is pathogen, which you can see on the slide which I have shared. Pathogen. Then the second side of that triangle is host, and the third phase is disease. Sorry, favorable environment. Environment. So all these three sides, all these three factors have been joined together, and then they cause disease. If all these three factors join together, then only you can expect a disease, not by a single one or not by the two of them. Say, a pathogen is there, but there is no host and no environment, then there will be ultimately no disease. And even if the host is there, if the pathogen is not there, then also no disease. And if both host and pathogen are there, but there is no favorable environment for the disease induction, then also there will not be any disease. Mere existence of pathogen and host will not cause disease unless this is favored by the environmental conditions, certain environmental conditions like air temperature, favorable soil temperature, soil fertility status, soil types, soil pH, and rainfall, relative humidity, soil moisture. So all these factors together, put together, they favor the environment. I mean, they constitute the favoration of the environment, and then they cause interaction between pathogen and host, and that is the very basis of the production of the disease or appearance of the disease. And again, pathogen should also be a virulent pathogen. If the pathogen is avirulent, it is not virulent, then also there will not be any disease. The pathogen will be fungi, bacteria, viruses, nematodes, mycoplasmas, viroplasmas, and so on. Similarly, the host also should be a susceptible one. The host may be present, but if it is a resistant host, there will not be any disease. So, in a very clear-cut sense, what we can define is a disease is interaction between a virulent pathogen, susceptible host, favored by climate, favored by the environmental conditions. So, if all these factors together they happen, then you can get disease. This is just to enlighten you what actually the scientifically disease means. Of course, all of you know, just to enlighten you or make you feel what the disease is technically mean, I just made this statement. Well, we know a lot of vegetable crops, you know all of this. vegetable crops we have. So instead of coming to the diseases, so the diseases majorly disease causing pathogens what we call. There are fungal pathogens, there are bacterial pathogens, there are viral pathogens, and nematodes as well. So all put together we constitute them as pathogens causing the diseases. Not just this, even mycoplasma like organism, spiroplasmas, and this is what you can say viroids. So they are all very less in number in terms of the disease causing diseases, number of diseases they cause. So generally, so what you can say fungal disease, majority of the diseases are fungal one, then some 5% bacterial ones, and viral diseases of course. Yeah, the recent trend is in last 10 years the number of viral diseases reported are being, I mean, the number is growing up like anything. So most of the diseases now, the new diseases are especially viral diseases, and of course nematode diseases. They are there. 2% of the diseases are only 2% of the diseases are nematode diseases, and maybe sometimes we sometimes fail to recognize the nematode disease because its symptoms are very slow ones, and sometimes they are not noticeable. And maybe that is the reason why nematode diseases are not that common. And maybe the type they cause, that means usually in bacterial diseases, viral or fungal, we say the period for the expression of the symptoms and death of the plant is usually very fast. In some cases, bacterial diseases, it takes within a day or two to the crop gets withered off, crop gets collapsed. Similarly, in case of fungal and bacterial also, fungal and viral also. But in nematode, see, sometimes the except one or two quick wilts and like this, so it is usually causing slow wilts, nematodes. So individually, nematode diseases cause take a lot of time to express symptoms and cause death of the plant. So that is what to say. That is why they are not being highlighted so much, but still they constitute a very important group of pathogens which cause the diseases. So with respect to the fungal diseases,
सर सर
yes,
सर आपका साउंड है ना इको हो रहा है थोड़ा सा, माइक के नजदीक आके बोलिए सर, थोड़ा क्लियर नहीं आ रहा है. हाँ हाँ यस यस
okay. Fungal with respect to the fungal diseases, अब आ रहा है क्या ठीक से?
यस आ रहा है सर, अभी क्लियर आ रहा है.
Okay, okay. With respect to fungal diseases, so there are number of pathogens which are causing fungal diseases. I have just listed very important ones. That is one Alternaria, Phytophthora. I mean, going in the clockwise direction, you can just see on the slide. Alternaria, Phytophthora, Peronospora, Ericifaceae, then Fusarium, Rhizoctonia, Cercospora, Sclerotium, Botrytis, Colutotrichum. So these are the major fungal pathogens. Normally, we see most of the fungal diseases are caused by these pathogens. Of course, barring these, there are again many fungal pathogens which are very less in number in terms of the diseases they cause. So similarly, so I have now I'll move into the group of pathogens. I have listed the number of pathogens in the previous slide. Here, particularly, I am taking here on the Alternaria diseases. Most of you, I mean, those who are involved in agriculture, at least diseases, you may have observed the leaf spots, a very common symptom, very common disease in most of the crops, almost all the crops which you grow. See, the Alternaria is one such pathogen. Of course, there are many which cause the leaf spots. So the Alternaria causes leaf spots in many of the crops, starting from potato, chili, brinjal, onion, carrot, cauliflower, broccoli, cabbage, radish, tomato, and the list goes on. So most of the vegetable crops and even the field crops are infected by this Alternaria. You might have seen in case of tomato and potato especially, we call it as early blight. Early blight. The name early blight means it usually appears early in the season, before the 30, usually at 30 days of plant stage. But here what you have to observe is identification of disease is more important. For every disease, almost all the diseases, there is a remedy except a few virus diseases. But thing is, you have to identify the correct disease. The symptoms, based on the symptoms, identification is very important. Here in case of Alternaria diseases, you can see, especially in potato and tomato, very typical. That chili also, see, you have a circular appearance of the diseases. It's like a target board appearance we call. See, the symptoms or the spots are having circular lesions, circular you can see, and inside we have a target like appearance. It's like a target board appearance. So circular rings are the typical symptoms in case of Alternaria diseases. So here potato or chili or brinjal, onion, so all those you can see the typical type of symptoms. Most probably these alternaria are highly influenced by the humid conditions. Most of them we see during the monsoon, yeah, I mean to say kharif season, wherein you have a rainfall, sometimes drizzling rainfall, and then high humidity and cloudy condition. And these are the very important conditions for the alternaria to sporulate and cause diseases and also cause in severe form. So these are common symptoms. See, of course, this is a pathological thing. I don't want to go in detail because Alternaria also, this is commonly what you say Alternaria in the under the microscope. And in case of course, there are again different species, which I don't want to go here because it is becomes a hardcore pathology thing. So mainly we are concentrated here on management of Alternaria diseases. I said, Alternaria is usually highly environmental dependent. Means if there is a drizzling rainfall, if there is a cloudy weather, and if there is a what I can say high humidity, these diseases will be severe form, in very severe form. In severe, it is so much severe that even after taking control measures, we will not be able to save the crop. That is the truth. So, if the environment is highly congenial, whatever spray you are going to have will not save the crop. And that is what the nature, I mean, with all ultimately, whatever we do efforts to save the crop, to protect the crop, ultimately it is the nature which has to join hands with us. If it doesn't, then it's very difficult. So here, of course, the recommendation, disease-free seeds, normally we need to have a disease-free, truthful certified seeds, which normally produced from the good mother plant, healthy mother plant. In seed treatment, normally it is recommended for all the foliar fungal diseases capped on at the rate of 3 grams per kg of the seeds. And avoid injury of the plant. This is a general cultivation advice. So usually if there are injuries, not only this alternaria, but it is commonly even bacterium also very much, I mean to say, enters into the host through the injuries made. So that will cause the diseases. And then limit leaf wetness water to the drip. So normally we have to go for the drip irrigation rather than the furrow irrigation, flood irrigation and all. Of course, nowadays farmers, especially in vegetables, product cultivation, mist commonly drip. So, but again, there the mist is also causing humidity. So that has to be taken care. So usually we have to take care of the humidity. Humidity should not be more than 80% if it is alternaria. If it is, I mean, if you can see the incidence of alternaria, then just you can lower the humidity if it is a greenhouse protected cultivation where you will have a control over limiting the humidity at least for some 48 hours. Remove or destroy debris after harvest. This is again a common thing. It is applicable to all the diseases because see the debris, those which are surviving in falling in the field, they are also serving as a source of infection. So normally the infections, these foliar diseases cause on the leaves dry up, leaves fall off and they stay on the field. So the pathogen in its dormant condition survive on such fallen debris and predispose, I mean to say, they serve as the source of infection for the next season. And of course, gift cultivation, this is ultimately one of the another, I mean to say, uh, another means of containing the disease. And of course, ultimately, the chemicals here, we have to go the least importance, but only during the inevitable things, inevitable condition, we have to go for the chemicals, especially the fungicides. I said in the beginning also, if the other conditions are, meant to say, environmental conditions are not supporting us, whatever may be the chemical you are spraying, that will not support you. And almost nowadays we are to switch on to integrated disease management. We should not attempt only for the chemical management. It is, of course, gives in to say control sometimes up to a certain period. But again, if other factors like if you you are not cultivating cleanly, if you are, to say, the field is fed with the debris of the previous season, so then it is of no use. So that is why, together with all the management practices, you can go for any of the chemicals here. Of course, the second one, mancozeb, have I told you? Mancozeb is a good one. It is a good old chemical, but it gives good protection. Only thing is, it is having some. If you are targeting for the, to say, export-oriented crops, then it is a bit difficult because it has a, what you can say, residual period up to 30 days. So, if crop is nearing harvest, you should avoid mancozeb, and then other hexaconazole and all, they have maybe some 15 days, what you can say. So only if you are targeting for the export production, you have to take care of the residual analysis, meant to say, not analysis, the residual interval or the pre-harvest interval, we exactly say PHI, meant to say pre-harvest interval. So till how many days of those harvest you should not spray. So that is the one thing. Otherwise, this mancozeb is a very good chemical. You can use it at the beginning of your season. It's a very good protectant. Then copper hydroxide also, hexaconazole also. Here you need to know the economics of the cost involved in spraying also. That also matters here, because see, there are again good number of chemicals which are costlier in nature are available. I have not listed. So this hexaconazole I have listed. So these fungicides which end with normally ZOLE, zole, belong to a group of fungicides, trizoles. They are called trizoles, and hexaconazole is one which is very cheapest. And further, after hexaconazole, we have propiconazole, tebuconazole, penconazole. So, like many zoles are there. So, all of them are a bit costlier. But if the, to say, if it is needed, so if the crop is very, very important, you are growing for a seed production or something like it's a very highly valued crop, then it is inevitable. You can go for the chemicals which are costlier also. But keep in mind that in the beginning, if the disease pressure is not that much, you may go with the cheaper chemicals. That will not only save your crop, but also, you can add, and when the disease severity is higher, you can go for again good chemicals which are costlier and very effective. So in the beginning only, if you go for the very costly and effective chemicals, then you will not be having the ultimate choices at the end of the crop season. So that those things are tricky ones you have to keep in mind while going for the management practices. The next group of diseases are polytrochum diseases, we normally call anthracnosis. I think you might have heard the word anthracnose, anthracnose of cucurbits, anthracnose of chilies, anthracnose of onion, so anthracnose of mango. So many crops, they are suffering from anthracnosis. And this anthracnose is caused by a pathogen called polytrochum, which is almost similar to Alternaria in its, way of causing the disease, especially the environmental conditions required. Of course, pathologically, of course, mycologically, taxonomically, it differs. That's different thing. But in terms of the environmental conditions required, the climatic conditions required for the development of the disease is almost similar to that of Alternaria. So that means to say, here also the drizzling rains, Kharif season, monsoon season, where the humidity is high, and intermittent rains, what you can say. These conditions especially trigger when to say the disease appears in very severe form. Like in cucurbits, chilies may be a very popular anthracnose. I think all of you have seen, those who are familiar with chili cultivation, or capsicum, whatever may be, chili capsicum. So this is a very familiar disease. You can see the photographs also. See, this is caused by colorectal, and you have here one more thing other than the alternate, that is the black dots you find on the surfaces, on the infected surfaces. You find the black dots that we call as acervillus. Of course, that is the, what you can say, sexual fruiting body, we call it pathology. So I don't want to go that detail. Only the symptoms you can see there at the capsicum. So it is also like a circular shape, but here what you can see is some black dots also you can see. So that is because of that fruiting body which is produced by Horticultural, and this is also causing severe disease in many of the crops, including the vegetables and many fruits also. So major vegetables, exotic vegetables, all are infected with this culturicum. So this is the different types and shapes of the culturicum seen across different species. So ultimately, here also management measures almost similar to that of Alternaria. Of course, deploying. Deploying is a common recommendation for any of the disease and even for the pests also. The very purpose of deploying, of course, it pulverizes the soil and nutritional, I mean to say, the soil, it improves the texture structure, but at the same time, with respect to the plant protection, disease and pest management, whatever the dormant structures they are there inside the soil get exposed to the sun, especially if the deploying is done prior to the hot summer, then they get exposed and thereby the inoculum that is already present will be reduced. So it is said if the initial inoculum is reduced, you will at least save 50% of the crop. So if the initial inoculum itself available for the infection is very, very high, then you can expect the severity of the disease. There is one mathematical formula also where we say x is equal to x0 e raised to rt. So this is the exponential formula. The very meaning of that formula is if it all the final disease is highly dependent on the initial inoculum that is available. Initial inoculum that is available. So you have to first target on reducing the initial inoculum, and that is through the deploying and then removing or destroying the debris and through the seed treatment. So all these are aimed at removing the initial inoculum. And then of course clean cultivation. This is during the time of cultivation. You have to have what you can say protected irrigation. So drip irrigation is preferred, and if it is a protected cultivation, you have to have your control on humidity. So you have to lower the humidity if it is exceeding more than 90 and the diseases are observed. And then the spraying again, almost the chemicals are similar to that of which I mentioned for Alternaria. So say, for example, carbendazim. This is many of you know that it is Babistin. Is carbendazim. It is available in many other trade names also. And mycochip also here, hexaconazole. So these three are the ones which are very cost effective and effective in the beginning. So you have to take care of, you have to note down these three chemicals which are very effective during the initial phases and very, very cost effective and which are helpful to reduce the inoculum at the beginning. But if the inoculum exceeds your control limit, then you can go for the triazole compound, which of course hexaconazole is also one of the triazole. And in the last phase, last disease, I told many chemicals like propiconazole, like this cresoxymethyl is also one of the chemicals. It is very, very helpful for the management of the anthracnose of many of the crops. That is at the concentration or at the rate of 1 ml per liter of the solution, per liter of water. So that will take care of the disease. Then the Phytophthora disease. These are some different type of diseases which are highly soluble and high moisture. Wherever there is a high moisture, that is these diseases take upper hand. High soil moisture, I need to say. So Phytophthora diseases are also important in vegetable crops and also in the protected cultivation. But here what you have to be, when the diseases are soil-borne, you should take extra care because they are surviving in the soil for a long number of years. If the diseases are airborne, like the previous one I told, Alternaria or Horticultural, so there the thing is, even if one season you find the highest, I mean, severe disease with some management practices, strict management practices, and good chemicals, you can manage and you can reduce down the incidence in the subsequent years. But if the disease is soil-borne, it is surviving in the soil, and whatever the amount of quantity of the chemical you are drenching in the soil will not suffice to contain the infection. Ultimately, what happens? So the next years, year by year, the inoculum will be built up, and you will end up with a severe disease, and almost your crop, your field becomes a sick soil. So that is why it is very, very difficult to know about these soil-borne diseases. So in tomato, you can see the Phytophthora diseases. In potato also, very typical potato. All of you know maybe late blight of potato. You know the very Irish famine. It is in the 1942 to 1945. 3 million people died in Ireland because of one tiny pathogen, this pathogen. So this in Ireland, the potato was a major crop, staple food crop, and the total potato was wiped out that time. And and they did not get potato from outside because of the World War going on at that time. So 3 million people literally starved and lost their lives because of this one pathogen, Phytophthora. See, just imagine how important this Phytophthora pathogen is. And then, of course, brinjal. See, all of them, these are highly infected by this particular pathogen. So these are, of course, the different sporangiosperms and the spores produced here. So here the main thing is, especially you have to look at the symptoms. I did not mention. So symptoms, see here you can see. Of course, in brinjal, I have seen shown that some root infection will be there, where you find. But typically, in case of tomato and potato, you can there is blackening or darkening of the what you can see dark patches, and starting from the margin of the leaves. You can see the margin of the leaves, dark patches, blackish discoloration, and that will extend up to the midrib. And especially on the corresponding lower surface, you find the growth, cottoning growth of the pathogen, mycelial growth, what you can say, cottoning growth of the pathogen on the lower side, the back side of the leaves, and that is a very prominent symptom to identify as what you can say Phytophthora diseases in case of both the potatoes and tomatoes. So with respect to the diseases management, see, I said again, deep flowing. So definitely, if it is a pathogen, soil-borne pathogen, deep flowing is must. There is no other way, because these will survive in the soil for a longer time, and you have to have deep flowing, especially prior to the summer, and even you can if the soil is something, the area is very small. Like if you are using for protected cultivation, meant to say very seed production area or very limited area, you can very well go for the even the soil solarization also. Of course, I have not listed here. Maybe in other places I will list. But soil solarization is also one good technique wherein you need to cover your soil after deploying and irrigating them. You have to cover it with a fine polythene mulch. That will be what it does. So that mulching will increase the temperature inside the mulch or plastic what is in that covering. So if it is priorly irrigated, so there will be wet heat generated, and that wet heat will because of the wetness, the pathogen structures they germinate, and because again immediately there is a wet heat, the pathogen structures cannot survive and they die off. So that is a very good technique, soil solarization. Raising the crop in the raised beds with the plastic mulch, of course, that is raising crop. Soil solarization is done before the crop being sown in the summer months. Then staging and removal of foliage and fruits up to 30 centimeter. This is very important with respect to Phytophthora. I said Phytophthora is a highly soluble, and if there are some the showers, heavy showers, then the soil splashes will also fall on the leaves, foliage that is very near to the ground. Even in the protected cultivation also, the direct effect of rains may not be there, but still the what you can say foliage very near to soil get infection. So that is why you have to remove the foliage that is up to 30 centimeter, and even fruits also, that will cut out the infection chain. And with respect to the management, the chemicals, again, I have listed very few here. You can even have find very big list, but it depends depending upon your requirement, your economics of crop production. So I have listed here first one, the Bordeaux mixture. You know, the Bordeaux mixture is 125, almost 130 years old Bordeaux mixture. Most of you know how the Bordeaux mixture is prepared. So this Bordeaux mixture is prepared out of the copper sulfate and lime. Of course, if you have any doubts in preparing, you can even ask me afterwards also. I don't want to go in detail about the Bordeaux mixture preparation. The Bordeaux mixture is one of the good chemicals still after 130-40 years for this type of pathogens like Phytophthora and all. And then copper hydroxide, mango chip again, metalaxyl amchud. See, metalaxyl amchud is one good chemical against this downy mildews and Phytophthora diseases. Then we have some combi products like cymoxidil plus mango chip. Then we have another crop that is dimethomorph, and then one more combi product that is phenomenon plus cymoxidil, and and even cresoxime to some extent. So these are all some very good chemicals which you can even go for spraying at a concentration 1 ml or 1 gram per liter of the solution. So ultimately here also this disease is also highly favored by the soil moisture, drizzling rains. These are high humidity. This is also high humidity disease. The humidity is this is especially seen in case of monsoons. They are especially seen in case of during the monsoon months. So this is one of the important diseases, and this is how the management is done. So then coming to the powdery mildews. Powdery mildews are also one of the important diseases. See, you might have seen this is especially in case of protected cultivation. Powdery mildews are really troublesome. The powdery mildews, on the contrary, I mean, other diseases which I dealt so far, they were usually monsoon season diseases, kharif season diseases. But it's powdery mildew and of course the rust, which I am going to cover in next slides. So they are the rabi season diseases, where the humidity, they don't require much humidity, they require dry weather. The rabi weather is very congenial for them. So the powdery mildews are also a group of diseases which also are very serious for the towards decreasing the yields. So they are seen in case of chilies, cucurbits, tomatoes, and not only the one mentioned here, in almost all the vegetable crops, even your aromatic crops, flower crops, all the crops, they are highly infected by this powdery mildew type of pathogens, powdery mildew group of pathogens, and okra also they are highly. So here, as I said, they are only the rabi season disease, mainly, not only, mainly, even in the kharif, sometimes if there is dry weather, they also do appear. So during the usually powdery mildew is what we say, the powdery growth. It's very easy to identify. Anyone of everyone can also identify. That is why the name powdery mildew. Because you see here, powdery growth, like it is, you see the symptoms on the crops or the host as powder is sprayed on the crop. So sometimes even on the upper side and lower side, both the sides, see, especially in case of chili powdery mildew, it is very difficult to eradicate because there we say in powdery mildew some ectophytic like things, mean to say ectophytic means if there is it attached to adhered to only the surfaces. If it is endophytic means it is even to the inner side. So that is why in case of chili is a endophytic powdery mildew pathogen, wherein it is in case of chilies powdery mildew is very difficult to eradicate. So these are some of the symptoms. So here in case of powdery mildews, you have to go for the crop rotation, removal of the infected debris, alternate host with some weed plants also, and of course the deep flowing. I have listed here, but it is not the one which is highly required for the control of the powdery mildew. Of course, it is also one of the component which is essential along with the management of other diseases. Increasing the air movement is also very important. So if there is a free flow of air, good aeration, then the disease will come down, and reducing nitrogen fertilizer. So usually nitrogen fertilizer also favors the disease. So there are, say, in case of powdery mildews, we if the it is difficult, then we have to go for the chemicals. Chemicals, it is very difficult. I mean, to say chemicals only invariably we have to go. I have listed sulfur second, but the sulfur is one which in the initial stages, excluding in case of cucurbits, in all other crops you can go for sulfur. In the initial stage, it is very cheap and very effective also if the powdery mildew incidence is seen at the early stage. And of course, then you can go for tridomorph. Then I have listed azoxystrobin also. And then in the bottom, triazole heading I have listed myclobutanil. It's very specific to the powdery mildew, myclobutanil. And along with that, you can even go for difenoconazole, penconazole, propiconazole, flucelazole, so tridamethone. Now tridamethone is nowadays is not available. You can just remove that one. But in barring that, all others are available, effective. But the one myclobutanil I told, it is very specific. 1 gram, even in some crops in the beginning, you can go for 0.5 grams per liter. So myclobutanil is very effective fungicide. So in the powdery mildews, we invariably need to go for the fungicides. And then we have this group of pathogens where you can find the leaf spots like downy mildews. Downy mildews in case of cucurbits, cauliflowers, cabbage, lettuce, and onion. So downy mildews are like Phytophthora. They are a group of pathogens. They belong to the same groups for Phytophthora and this downy mildews. So cucurbits you can see, but the symptoms are a bit different. Here in case of downy mildews, on the upper surface, you find these type of lesions, yellowish lesions, yellow halo, yellowish, and on the corresponding lower surface. On the corresponding lower surface, you find the mycelial growth or the cottony growth of the pathogen, and that's very typical here in almost all the crops. So lettuce, even cauliflower, cabbage. So it's downy mildews. These are also highly moisture-loving fungi, like Phytophthora. If the intermittent rainfall, drizzling rainfall, or continuous rainfall, or high humidity, these will cause. And moreover, it is also sometimes if the night temperature is if it is very low, then that is also very congenial for the development of this downy mildews. And downy mildews are if normally if they are very congenial, the environment is there, they take the upper hand and they will cause 100% losses sometimes. But they can be managed with the, especially in case of protected cultivation. If you reduce the relative humidity and if you give the more air circulation, it is definitely can be manageable. Unlike it's not like that a soil-borne disease. It's not so. It is also soil-borne, but not that like wilts. So here, what I can say, integrated management practice is very important, like deep flowing, because it is also a debris and the inoculum is in case of soil also. In the soil also, crop rotation and destroying the culp piles and feed host. So usually, if there is a piling of the culled ones, then that has to be destroyed. Otherwise, that serves as a source of infection. Of course, definitely disease-free seeds and transplants we have to always focus upon. Bodo mixture, see, for the downy mildews and for the Phytophthora. So, Bodo mixture is very useful, though it is a very old recommendation. And then the chlorothalonil, mancozeb are next step of the recommendation, copper hydroxide. But metalaxyl amger, you can note down that downy mildews. Metalaxyl amger is a very typical, very specific chemical for the downy mildews spray. And then dimethomorph, phenamidone are all new generation chemicals. Metiron plus the pyromostrobin, formoxidone and suboxynol, iprovalicarb and propinep, and drazoxymethyl. These are also recommended. These are very costlier ones. You can what I expect is you can go up to metalaxyl amger, and if the crop is very important, highly economical, then you can go for the below mentioned 4 to 5 chemicals. Only thing you have to take care is same chemical cannot be repeatedly sprayed. It will also influence on resistance development. So maybe alternative sprays of these chemicals will give good protection. Here, this is fusarium wilt. This is highly soluble disease. Wilts normally. Maybe if there are 4 types of pathogens again causing wilts: fusarium, rhizoctonia, sclerotium, and even the bacterium, and even nematode also involved sometimes. So these wilts are very dangerous diseases because they are surviving in the soil for a longer period of time. You can take of chili, tomato, cucumbers, and we call it as they are polyphagous in nature. They cause infection to almost all the cultivated crops, vegetables, fruits, field crops, cereals, pulses, everything. Yeah, infected by these fusariums. So with respect here, vegetables, especially all these vegetables which I have listed, they are infected. So the normal symptoms are, you can see the pale yellowing or pale appearance of the leaves in the beginning. Then we say the leaves droop. In case of brinjal, you can see drooping. That is, they lose the turgidity and they are just drooping. So that is the symptom. And then ultimately, wilting, which leading to death of the plant, complete yellowing, death of the plant, and then drying of the plant also you can see. And such plants, when you uproot, in case of okra, I have shown that, you can see the photograph. See, when they are uprooted, you can see the root system is damaged. Roots are blackened, discolored, and even when you take out the, what you can say, discolored roots, if you just stems, you can take a section, you can see vascular discoloration, also see the So this is the typical symptoms of the fusarium builds, and these are of course pathological observations where you can see the management of sorry, this is these are the conidia. I'm not going to so with respect to management, as I said, it is highly soil borne. So you have to invariably go for a deep summer flowing, follow crop rotation because they are going to survive for a number of years. At least 2 to 3 years of crop rotation is must if your field is infected with fusarium wilt, or if you go on planting same crop like tomato in the protected also. If you are going planting tomato after tomato, potato after potato, or any vegetables like some chili, brinjal, anything, then you will end up with multiplying those pathogens every year and higher incidence year by year. So we have to have crop rotation. Then, of course, the disease-free seeds, then the chemical recommendations, the seed treatment, even carbon disease also, and avoid sowing the temperature side. Here, I would like to stress upon one management aspect, that is seed treatment with trichoderma viridae or even application. So trichoderma, I think many of you know, and in the next point also I have listed Pseudomonas fluorescens. Both of these are biocontrol agents, and biocontrols are working well with respect to the wilts especially. Of course, nowadays biocontrols mechanism is reported in all the diseases. There is a recommendation of trichoderma. You might have knowing that trichoderma is commercially available. It is a fungal bioagent. It is a fungus basically, whereas Pseudomonas is basically a bacterium. Pseudomonas is a bacterial bioagent. Trichoderma is a fungal bioagent. So these bioagents are highly commercialized, popular, and they do have real efficacy, especially in case of wilts. So trichoderma viridae, sometimes it is recommended. I mean, recommendation is there to go for seed treatment at the rate of 4 grams per kg. Pseudomonas at the rate of 10 grams per kg of seeds. But even you can, in the next slide, I have told that 2.5 kg per hectare with 50 kg of FI. This is a very good method of applying the trichoderma, especially. See, what we can say is enrichment of FIM with trichoderma, we call. That is, you have to have the commercial formulation. So again, one thing you should keep in mind is whenever you buy a commercial formulation, maybe from public or private, doesn't matter. The thing is just try ensure that it is freshly prepared. Sometimes some private firms they give expiry data up to 2 years, but it is see it's not work well. We might say 2 years means here they have to the storage conditions also matter. We have to keep it that low temperature. But I'm not sure whether all the dealers are going to keep in the same conditions, in the same temperature conditions. That is why what I blindly recommend is ensure that it is freshly prepared, not later than 3 months. So that trichoderma you should apply. I mean, you should mix up with the FYM at the rate of 1 is to 10. Here I have 12.5 kgs in 50 kgs. It is just like 1 is to 20. Of course, it doesn't matter. So this FYM, you have to mix it with this trichoderma FYM, and you have to shade dry for at least 15 days. If it is 1 month, it's very good. So this trichoderma uses that FYM, finally decomposed FYM as a base, and that will grow, multiply in that FYM. And this enriched FYM with the trichoderma is you can put directly to the field before the what is the preparation of the field for the sowing, or even you can apply to the hills directly in case of chilies, tomatoes, and brinjal like crops. So this is having more advantage or more efficacy of a trichoderma application. You can get it through this type of application. This is the thing which I wanted to stress upon, especially in case of wilts caused by fusarium or rhizotonia, or even in case of Alternaria also, and sclerotium also. So, which sclerotium and rhizotonia I will deal in the next slides. Of course, and then the next spot dredging with trichoderma. Suppose, see, thing is, you cannot expect a 100% control in biological control, and immediate control is also not possible. If this is in advance, you have to apply. Okay, but with respect to the sudden measures, if your crop is highly wilted, then you can go for drenching with carbamazepine and even copper oxychloride, also COC. I have mentioned that 3 grams per liter. That is drenching at the root zone. But here again, the question is how economical it is. If your crop is infected to the extent of 5 to 8%, then only you can go for drenching. How not only that affected plants, even the plants surrounding them. Not all. If you drench a whole field, it is economically not at all feasible. So you cannot drench whole field. Only the infected fields, infected plants, and the surrounding plants. If it is that is what. So that is why in protected cultivation also you can go for trichoderma commercial application. Commercial formulation of trichoderma in a FYM incorporated mode, so that it is very beneficial for you in future. It is also against the weeds, not only the future weeds, but against all the weeds. Okay, please keep in mind. So this is the one. This is also a rhizoctonia disease I was telling. This is also causing wilts. Here the difference in fusarium and rhizoctonia is that this is also wilt causing pathogen, but typically you see bark peeling. The major symptom here is, of course, above ground symptoms are very common: yellowing, drying, drooping, everything. The typical characteristic symptom is here, that is one which can differentiate from fusarium, is when you see the bark gets peeled off, sloughing of the skin, what you can say, the outer layer. So that is the major typical symptom here. So rhizoctonia. But other than that, the control measures and all the things are similar to that of fusarium. There is no extra control measures. So all the chemicals, biocontrol, everything. Even you can go for this deep flowing, right from the deep flowing up to the chemicals, use of bioagents, everything is common. I'm not going to repeat it again. So I have sufficiently elaborated while dealing with the fusarium, and the same thing can be repeated for the rhizoctonia also. One more disease is gummy stem blight. It is especially in case of cocoa beets, and nowadays it is seen in more severe form in Karnataka, especially I have seen, but I don't know in other states. See, this gummy stem blight is caused by Didimella rionia pathogen. So you can see stem, oozing, gummy oozing on the stem, you can see this disease. So similarly here also, we don't have any special, like in case of here also we can go for the bacterium and pyramidostrophin, this one chemical. Of course, that is in very small quantities, and there are again some more diseases which are purple blotch, specific to some crops. This purple blotch in case of onion, stem phyllium leaf blight in case of onion. Okay, so this is other diseases also. Here, other foliar diseases also. Here also you can go for. I think that we are running short of time, so almost 50. Okay, I'll go a bit fast. Sclerotium diseases. Sclerotium. Here also this is a root disease, wilt disease, like things. So here you can see here this root system, or on the surface of the roots you can find mycelial growth, like in carrots, even in chili. See, on the soil surface also you can see mycelial growth. In onion, in carrots, in potatoes, so all the crops are infected with sclerotium. This is also a disease which is soil borne, and it is highly surviving in the soil, the inoculum. So for this also management, whatever the management I told for the fusarium holds good for rhizoctonia and sclerotium also, because all the three are soil-borne pathogens. There is trichoderma application through the FYM incorporation, and the chemicals, deep flowing, crop rotation, everything is similar. Okay, so these are cercospora diseases. Of course, it is foliar disease like Alternaria and fusarium. Alternaria and cultivation, I told in the beginning, like these diseases. So here you have on the chilies, you can see the very typical symptoms. Here, the margin of the spot is dark and the center is gray color. You can see in chili, very typical. You can see in coccinia, below figure. You can see in pallor. See, the margin is dark and the center is grayish white, gray color. So this is a typical cercospora. And here also the management practices I don't want to go elaborate, like Alternaria, whatever sprays I told, that you can see. So it is the causation, the climatic conditions required, and management. All are similar to that of Alternaria and Polypodrica. Okay. So the botrytis diseases, these are the ones which appear in case of during the fruiting stage, and then there is a very heavy rainfall. So this is like something causes rotting of the infected surfaces in case of cabbage, also in case of tomato, also cucumber, beetroot also. And these are not very common. Sometimes if there is a rainfall, especially during the fruiting stages. So here you can go with the pre-harvest spray because this is even causes infection after the harvest also. If the infection is there in the field and after the harvest, if there is a humidity, like if the moisture is there. So normally during the monsoon season or the kharif season, if there is some rainy season, during that if the harvest is taken, then there is a moisture is there, or even with the respiration also, the harvested product respires definitely. So due to that also there will be buildup of the humidity and that will also cause the diseases. So this to avoid that, usually we can go for pre-harvest spray. Only thing is during the pre-harvest, if you take care of other diseases like Alternaria, Bagalkot, this disease automatically can get taken care. And then bacterial disease. Nowadays, bacterial disease is also very important, especially in case of chilies and tomatoes, where the bacterial wilt, the very first photograph you can see in case of chilies, bacterial wilt is more dangerous than the fusarium wilt. Because see, bacterial wilt immediately after showing the symptom, within 28, 24 to 48 hours, the whole plant wilts. And that is how you can differentiate between the fusarium wilt and bacterial wilt, fungal wilt and bacterial wilt. In case of bacterial wilt, if you see the symptom today, within 2 days, the whole plant gets dry. Being green itself, the plant gets wilted. But that is not in the case of Fusarium or Rhizoctonia or Sclerotium. The plant turns to yellow and slowly starts wilting, and the whole process takes 15 days. But in case of what you can say bacterial wilts, the Solanaceous crops especially, the wilting is very fast, so much so that the plant before getting into yellow, it will wilt. That is we call green wilting. So that is one thing. So here also in case of with respect to the management, we have to have if the infected crop is there, next year we have to go for this rotation and then the seed treatment because we don't have much bactericides. Only streptocycline along with COC is the only one combination which serves well. But if the disease appears, then it is very difficult. Even recommendation of drenching is there, streptocycline the COC, but it is very highly costly. It is very costly. So cost efficacy is also one of the things which is worrying. Foliar application and drenching is also recommended. That is why have to be very careful that seed treatment is very good. And then the control over irrigation. If the irrigation, if initially you off or else there will be spread of the disease between the. Sir, my voice is audible?
हाँ सर, audible sir.
Okay. So it is okay. So then quickly move on to the viral diseases. There are different viruses. In case of tomato, big bud virus, tosco virus, and in chilies, leaf curl virus. It's a big story since many years. So the since I mean, even though scientists could be able to able to release at least one resistant variety because the resistance is not seen wherever anywhere. So chili leaf curl virus is problematic all the time. And chili, you have many viruses nowadays. Every scientist is reporting one or the other viral disease. So it is molecular biology has made an intervention, intervention here, and every now and then, every day you will seeing a new virus is reported. But ultimately, this chiefly the curves in the Horticultural viruses are the ones which are very dangerous. And viral diseases don't have any remedies. Once they are infected, we don't have any remedies except uprooting and burn. So that is why prevention is better than cure here, because most of the viral diseases they are transmitted through the insects like the jar seeds, leaf hoppers, thrips, mites. So the major focus here managing the viral diseases is to have a chemical space, either imidacloprid as a seed treatment to for the sucking pest control, or sometimes even after the sowing, because that imidacloprid seed treatment can give production up to 30 days. And after that, even if there is a sucking pest incidents like thrips, mites, leafhoppers, acids, whiteflies, if they are seen, we have to invariably go for a very good chemical, especially the insecticide for the management of the insects, which are especially for vectors. And one more thing, that's border problem. Border crop is highly recommended for all the vegetable crops, especially when they are highly prone to viral disease. The very mechanism of border crop is that is we have to have a border crop like with the help of with the, sorghum or bajra, any tall growing crop. If it is if you are able to in the open cultivation, the protected ones, of course, there is you have nylon nets and other protections. There is no problem at all. You don't need to go for the border. But it is openly cultivated, you have to go for the border crop. That is just to hinder the appear, I mean to say, the movement of the virus, so the vectors of the viruses. And same case with the productive cultivation also, we have to have a keen observation on the sucking pest. If these sucking pests are noticed, we have to immediately take care of them. Normally, these thrips, mites, incidence is more in case of productive cultivation because the conditions are highly congenial. They not only directly damage the crop, but also indirectly causing the viral disease. So, especially in chilies, capsicums, they are highly problematic. So, of course, here you have seed treatment in the but I said already, growing the nursery under nylon net, meshes, soil application of Furadon, and of course, seedling deep in case of imidacloprid. These are all insecticides which have been strength, especially for the insect control, vector control. And the last one, there is nipping of seedlings during transplanting, in chilies especially. Especially chili, if they are transplanted and if they are sown also at 30 days stage, nipping is just to remove the top leaves. Especially in case of transplanting also, while from the seed nursery to the main field, if you are transplanting, you can nip, that is just to take off the upper leaves. That will, what happens there, the usually the viral incidence or we can say leaf curling is seen on those top leaves, and that will be taken care. So the population of insects, I mean, the sucking pests that are going along with the transplants will be less, so that in the main field or the transplanted field you will have a less infection. So dipping especially is recommended in case of chilies for the management of the viral diseases, especially the leaf curl viruses. So, of course, integrated management is there. So, important of this is especially in case of open cultivated crops. Removal of early infection, spraying of insecticides, neem kernel extract, mulching. So, these are all in case of the open. And in protected cultivation, we have to invariably go for the sucking pest management. Nematode diseases. So nematodes typically they cause the root disease. So on the roots you find the typical nematode symptoms. You find galls, but most of the times, especially in protected cultivation, the nematodes are the big problem. But we fail to identify the problem. That is because you find the host plant which is showing it is just yellowing symptoms, paleness, but most of the times what we say is it may be because of the lack of the nutrition or some fungal disease. But especially you should look at the roots. When you see the roots, you find it the knots or the galls. That is definitely a root knot nematode. In nematode, we have a lot of species, but the root knot nematode, that is Maleidogyne indica. So that is the most widely abundant disease causing nematode, and there are many species. So normally in case of nematode, if you ensure it is a nematode disease, you should immediately take care of. So here is a disease also, high disease, soil bore, big growing, or that is soil management is must. Crop rotation, it is as good as what you can say the fusarium and all. Intercropping with marigold. So normally in case of nematode management, we recommend marigold as intercrop or as something along with that we can grow. This is the antagonistic crop, that is the root exudates of marigold are harmful for the nematodes. So that is why marigold is recommended as an intercrop. Of course, application of neem cake, it during the process of decomposition, it produces some of the volatile chemicals. Those are harmful for the nematode population. And then nematicides we have for it. Then carbofuran, of course, there is from 2022 there is a ban on carbofuran for it, but still now they are available. But you can go for it. They are very good nematicides especially. Then if you are going for the biocontrol agents, for the nematodes especially, there are two biocontrol agents that are Bacillomyces and Pasteurium panavitis. Bacillomyces is a very good one. Bacillomyces, this is a very good one. This is a fungal biocontrol agent. That is a fungus. It is used for the biocontrol of the nematodes. So this is the one which you can go. So these things you have to take care with the nematode. So in the addition, so it is just in a nutshell, you have good planting materials, description of crop debris, tillage, crop rotation, intercropping, mulch. So all these things which are just in a single slide, I have just summarized here. So fungicides, of course, it is biocontrol agents. So I thought of giving much stress on biocontrol agent, but anyway, this slides and in notes it is there because it is. I think I am already have taken more 1 hour, so half an hour is left. So many exotic vegetables have to cover. Lettuce is much so these exotic vegetables. So lettuce we have septoria spot. Here I will go a bit fast because I have already in case of generally covering vegetables, I have told the groups of diseases. Here specifically I am going for the symptoms and very specific thing what I will deal with. Okay. So in case of lettuce, here septoria spot. See, this is like a cercospora or other alternative spots. You have symptoms like this patches of white grayish pin-sized symptoms, and of course management here is concerned. This is of course pathogen-free seed. You have to have a good healthy material, planting material. Of course, everyone will take care of that. But again, when it is planted and if there is a rainfall, then normally such diseases are seen. Septoria you can even see on tomato also. So one good chemical, like here I have listed copper hydroxide, the last line you can see. Similarly, the Alternaria, in case of Alternaria, very first disease which I mentioned. So the chemicals like mycogen and one hexaconazole I mentioned, same chemicals also refer here for the septoria management. Then the downy mildews, so here also you can write downy growth. I have shown downy mildew symptoms of lettuce in the previous slides also. So on the upper surface you find yellowish, and on the corresponding lower surface you find mycelial growth. So here one good chemical I have already mentioned for the downy mildews, metalax char. Other than that, I have mentioned all other integrated management practices that is right from the deep soil because this is soil fungus. So sclerotinia, of course, about which I have not covered previously. Sclerotinia is one such pathogen which is infecting during the hot days also, and here the rotting causes from the roots to the head, from the root to the head. Sclerotinia, the white cottony growth or cottony mold is developed on the rooted tissue, and it's a very hard tissue, becomes a hard the infected portion, and this fungus can survive up to 8 to 10 years, 10 to 10 years. And this is since it is soluble, you have to take care of the sclerotium diseases. What I have explained for the sclerotium diseases, crop rotation, deploying, application of pesticides like COC. So all those things hold true for the sclerotium rot. And then the bacterial soft rot, which is caused by bacteria. So this is more of a post-harvest disease. But only thing is you have to have a good pre-harvest management also. Disease-free seeds and during your crop growth in the field or in the protected cultivation, then you have to have one spray of streptocycline and COC to ensure that there is no bacterial growth. So the sometimes even after usually it appears after the harvest only on the surfaces if there is any what is like wound like thing. So especially in case of bacterial diseases, especially the post-harvest diseases, they appear if the product is not post-harvest product is not handled properly. If there are any subsurface or superficial damages also, that will lead to bacterial damages. So those things to be taken care. And then this is a again one more disease. Sorry, the previous one. So with clearing, so this is also in case of you can see the viral diseases. With clearing, use disease seeds and the seeds. The powdery mildews. Powdery mildews, of course, productive cultivation is a big problem because the conditions they help for the disease development. So humid weather and even dry conditions also. Powdery mildew fungus can survive in the wide range of weather. So only thing is air circulation. Of course, chemical here I have listed hexaconazole, but air circulation so is very must. Then lettuce big bean. So this is a disease. This is lettuce lettuce leaf infected with the big mosaic virus. So this virus is transmitted by the fungus So these are the so only thing is we of course the virus diseases we don't have any management aspects like spraying or anything. Only resistant varieties if available, but actually they are not available here. Yeah, only just for information you can take this one. So in case of celery, we have the Cercospora. Cercospora same management. I have told about the Cercospora leaf spot previously also while listing that Cercospora leaf spots. This is the typical symptom, and you can go for same follow the management practices which I told previously. And with the Septoria, I told in case of lettuce also, same Septoria, these are the diseases. So same management practices we can follow here. Then the Cercospora again, same management practices. These are the symptoms you can see here. Damping off. Okay, damping off, I did not use this word. I would have actually told in case of vegetable diseases. Damping off is especially a big problem in the nurseries. Damping off is especially again soil-borne pathogens like Pythium, Phytophthora, Fusarium, Rhizotonia. So many pathogens are involved in it. Wherein you can see pre-emergence damping off and post-emergence damping. We say in case of tomatoes and chilies, especially in case of solanaceous crops, they are very problematic. Crops, when seeds after sowing, sometimes not able to germinate come out. When you see just inside the soil, so you see the seeds are rotten. Just the plumule radical has not come out. And sometimes even after the emergence, there will be slight constriction at the collar region, a blackish discoloration at the collar region, and the leaves, sorry, plants, birds plants will fall off. So that is what we call post-harvest, not post-emergent damping. Post-emergent damping. So for the damping of, you have to have a raised seed beds and a very sandy soil. There should not be any water stagnation. There should be free flowing. Water should be free flowing. Of course, the seed beds require more water, but water should not stagnate. That is the one which is a major predisposing factor. The stagnant water to ensure that the seed beds are sufficiently raised and they are with the free flowing water without stagnation. And of course, here you use biological control managements like trichoderma and pseudomonas. If you want to go for the chemicals, you can go for the carbon disease drenching. So then anthracnose, of course, in celery also, same thing. I have explained enough about the anthracnose in the previous slides. The same things we can apply here. So white mold, these are also sclerotium diseases. Sclerotinia rot, I told in case of lettuce. So same things holds good here. Juchini, crop, powdery mildew. Please just see the slides because all management methods are same thing. In case of fusarium also, fusarium wilts, and this is downy mildew of Juchini, and downy mildews. This is a bacterial blight. Okay, we can go for streptocycline and copper sulfate mixture. Alternaria diseases I have explained sufficiently. Then this is another foliar Plextochlorium. Same Alternaria management you can follow here. Phytophthora, and then okay, this is another what you can say foliar disease where you can just follow the symptoms of the Alternaria, sorry, management practices. Verticillium is like fusarium only. This is causing wilts. Okay, this is mostly in Indian conditions they are less effective. In the temperate crops they are very severe. Sarcospora and parsley, the another exotic vegetable. Here you have Alternaria problem, and here of course you have this Alternaria infection photographs. Bacterial leaf spots follow the same things which I told for other vegetables. Then these bacterial symptoms, you can see bacterial symptoms again. Blight in the younger leaves. Bacterial, so this is another bacteria, Erwinia. So two Xanthomonas, Erwinia. So they are both of them are problematic here. See, basal soft drop, you can see in case of Erwinia causing this disease. And in above ground you find collapse or bleaching of the parsley foliage. So in bacterial disease, we have to have to say drenching, but if it is only in protective conditions, okay, fine. A highly economical, you can go for. Otherwise, for normal crops, drenching is not recommended. It is highly cost. Botrytis, I told botrytis previously also. So plasma, this is downy mildew, and then this is powdery mildew, upper surfaces of the leaves. So I'll go quickly. Many slides are there still. This is development. This is the rhizoctonia, root cord causing production. Phytophthora, again, specific here in case of the parsley. Phytophthora rots, you can see here. Collapse of the plants. A rapid wilt. So sclerotinia rots, septoria, root knot nematodes. I have told in case of tomatoes, field infested with the nematodes, parsley especially. So very stunted growth, very pale appearance. It is looking as if it is devoid of the nutrition. And artichoke, the next crop. So here we have curly dwarf virus, viral diseases, bacterial crown rot, botrytis grey mildew. In case of kale, Alternaria leaf spots, anthracnosis, damping offs, downy mildews. In case of red cabbage, we have Alternaria leaf spots, management measures, brassica rot, black rot. So this black rot is especially in case of cabbage, you might have seen this is one important disease which is caused by Xanthomonas, a bacterium. So this has to be managed with the same chemical, streptocycline plus copper oxychloride. Streptocycline 0.5 grams, copper oxychloride 2 grams. The both mixed together in 1 liter of water. This is a very good recommended mixture for the bacterial diseases management. Same thing, I have the very first seed treatment and even spray also. Streptocycline 0.5 grams, copper oxychloride 2 grams, sometimes even go for 3 grams also per liter. If it is to be seed treated, then you have to take it for 30 minutes. Then brassica downy mildew, management cabbage soft rot, capsicum, in case of capsicum, damping offs, anthracnosis, powdery mildews, bacterial leaf spots, leaf curl, mosaic, in case of turnips, Alternaria leaf spots, club roots, sclerotinia. I have all explained in the manual what they are supplying. See, I have given sufficient information in like a word format that also you can read out. Turnip mosaic, and of course, very in nutshell. So, what are the avoidance principles here? Avoidance, exclusion. You have to avoid. You have to exclude the pathogen. You have to eradicate once it is into the field. You have to protect with the chemicals and the resistant varieties if they are there. You have to go for. So, what is integrated disease management? You know, all of you just have the information is there. In a nutshell, I tell you, we have many types of management measures like a chemical, cultural, resistant varieties. So all together, put together will give a integrated together will give a idea what the concept is integrated disease management. So most of the times we normally depend on a single measure that particularly spraying of a chemical. We target only that. So that is not at all feasible. First thing, it is highly costly. Second thing, only if you are depending on the chemical, the disease cannot be controlled because other things will add to the inoculum, and mostly if there is an exportable crop, you will end up with the residues. So nowadays, residue-free, good, healthy, safe production of the vegetable is highly aimed, and in this we are highly discouraging the chemicals, and only you have to go for the integrated manner disease management. So especially protected cultivation, what has to be followed that I have given here. So in case of greenhouses, classes, how you have to maximize the yield and minimizing the diseases. So it is all there. Only you have to check the control entry of that one. You have to ensure the hygiene, starting with the disease plants and growing the environment, control the growing environment. This is the most advantage for the protected cultivation. You can have a control environment in your hand. You can control the growing environment. That is the one advantage, high advantage you have in the protected cultivation. And of course, inspecting the plant regularly, waste management because they will dispose the diseases and pests also. control of insect and waste, and fungicide. The last choice is fungicide. So these are the mantras or the principles we have to have in case of product and cultivation. So all those things I have neatly dealt, and it is in the word format. So it is almost 1.5 hours. Just a minute. So that's this each thing I have explained here. Each what I said mantras or the principles I have explained why it is to be done and what is to be done. Okay, so fungicides are the last preferences and cultural practices. I have summarized what I have told so far. Those things are summarized. Physical and biological control. Much importance is this biological control nowadays. And of course, viral disease, we don't have any management measures, and we have to have three approaches here, aiming at the root environment, viral environment, and reducing the vector environment. So, by this using three items, we can reduce the viral diseases. And once viral disease is established, we don't have any management measures except uprooting and burning. So this how you can manage aerial environment, regulation of temperature, relative humidity, ventilation, water droplet sizes. So all this will add in advantages for the protected cultivation. So fungicides and all only at the time you can just mention have. And the vectors are the next one using the nylon nets, yellow sticky crops, insecticidal sprays, neem cage, maintaining the hygiene. This way you can. So still, in spite of this all what I have included the information, there are still problems. Especially there is a lack of available chemical control measures for that. We don't have a special research for greenhouse condition. Whatever that is being done for the open cultivated crop, and that has been borrowed in the greenhouse conditions. No special research has been done, even especially in India. So that is the one lacuna in case of protected cultivation. There is problem of frequent harvest over a long period. So you have a crop for a longer period, frequent harvest, that will disturb the environment and that will add to the diseases. And then the third is non-availability of equipment, especially pesticide. So we don't have a special equipment, special thing for the protected cultivation. Nowadays, of course, the precision farming, the sensors-based sprays, they are also planned, but practicability is still a question, and they are highly costly also. So we need to develop certain equipments that is especially for the protected cultivation. Those things are not with those are the research gaps. Maybe in the years to come they will be addressed. And I think with this information I end my talk here. Thank you for patient hearing. If you have any doubts, you can clarify here also. And even you have my mail ID. I hope the organizers will give you my telephone number is also you can note down, and you can call me anytime if you have any questions, any queries. You are all welcome. And with this, I close my whatever I wanted to present. Thank you.
Thank you, sir.
Thank you, Mahesh.
I request to all participants, if any question regarding this lecture, please put up in chat box or raise your hand. Thank you, sir.
Okay,
I
so
think
somebody has said ask the PPTs.
okay.
Anyway,
So,
PPTs and presentation are all available to you.
yes.
And you can even note down my phone number. Here
Note down your number.
I have posted my phone number if you
Yes.
have any doubts, and even my email ID also if you want. So if you have any specific questions or something to be discussed, kindly you may send a mail or you can call me. The same number is in WhatsApp number also.
Okay. Thank you. Thank you, Dr. Raghavendra Mehta, sir,
Thank you, Mahesh, for giving me the
for such a nice
opportunity.
presentation, a very informative lecture to integrated disease management of protected cultivation. All topic covered in exotic vegetables, all type of vegetables, and disease, fungal disease, and all these and control measures. Thank you very much.
Thank you.
Uh, I would like, on behalf of, I would like to thank uh, Dr. J.S. Pachurke Madam, Organizing Secretary of this training program and co-principal investigator of CAST project MPQ Rahuri. And I would like also to uh, I would I would also like to thank Dr. Sunil Gorantiwar sir, principal investigator of CAST project, and also thanks to all participants, research associates, faculty members, and with the permission of organizing secretary, I conclude this morning session. Thank you, all of you. Thank you, sir.
Thank you.